To continue with the day, we have Dr. Vijay Shankar Ashokan to enlighten us on the topic microscopy and bioimaging. Dr. Vijay Shankar Ashokan is a senior researcher at Chalmers University of Technology, Sweden. He obtained his PhD in physics from the University of Bergen, Norway. He is having a 15 years of research and industrial experience from various institutes like Indira Gandhi Atomic Research Center, Kalpakam, and Indian Institute of Science. He has strong work, research, and experience from four international universities, including University of Oslo, Norway, and Zhejiang University, China, and having the strong research collaboration with five universities and colleges from India, UK, China, and Sri Lanka. He has published several research and review articles in reputed international journals. We are awaiting to listen your inspiring words, sir. Here comes Dr. Vijay Shankar Ashokan. Over to you, sir.
Yeah, thank you. So I just try to share my screen.
Yeah, sir, definitely you can share your screen, sir.
So let me know if the screen is visible and
No, sir.
is the presentation mode is visible now?
Yeah, now it's clear, sir. Now it's
Okay.
clearly visible.
So very good afternoon to everyone. I think it's good evening in India, right? Yeah. So it's pleasure meeting you all here through the Google Meet platform. Today I'm going to cover the microscopy and bioimaging. Is my voice is audible, right? Is it clear?
Yeah, it's really clear, sir.
Super. Okay. Let me just try to cover basics of microscopy and what we are doing with microscopy in respect to the bioimaging or to the biomedical application in perspective, in particular to the materials perspective. So because basically I'm from material science background, I do work with the material characterization, I do work with electron microscopy of advanced techniques. I have wide experience around 13 years of microscopy alone with different universities around the world. So I just try to cover the basics of microscopy and with bioimaging at some part. And I try to give you some input on what are the advancement what we are into the microscopy and into the bioimaging for the future need. So far we know there are applications of material characterization through microscopy. In addition to that, in a present modern world, we are into like machine language or artificial intelligence for the imaging uh, or imaging enhancement, we can say. So I just give you some input on that. I try to cover as much as possible within this 1 hour. So I'm basically now sitting in Gothenburg, second largest city in Sweden within the Scandinavian countries, one of the beautiful city in the Scandinavian country. Um, Volvo is headquarters in Gothenburg. uh, Volvo bus, trucks, and cars. I basically graduated from Coimbatore Institute of Technology and post-graduation from PhD College of Technology. Did some part of my postgraduate research in Indira Gandhi Atomic Research Center. Then later on, I carried out part of my research work in Indian Institute of Science, Bangalore, in Materials Research Center, where I was introduced with a microscopy. I was a scanning electron microscopy operator there before joining my PhD in University of Bergen, Norway. I did part of my PhD in University of Oslo with short-term grant, around 6 months in Oslo University. Later on, I continued started my first postdoc with Zhejiang University, Hangzhou, where I was working with electron microscopy team. That's my first and very beginning towards the microscopy world. In particular, to the only with the microscopy world, I can say. Before that, I was working with material characterization and microscopy was part of my work. But from Zhejiang University, I was completely become as a separate microscopist, or we can say individual microscopist. Then I continued my microscopy work in Chalmers University, Gothenburg. Now I'm almost a senior researcher here. I have developed few collaboration around the world, including UK, Exeter University, Swansea University, and Western Norway University of Applied Science. through the Indo-Norwegian collaboration project, we have developed part of good collaboration work with Coimbatore Institute of Technology, and it's now extended with PSG and University of Jaffna, Sri Lanka. Rest of the universities like Madurai Kamarajar, Pondicherry University, and Zhejiang University, I have few collaboration in terms of publication. Of course, there are newly added university, that's SRM. We recently published 2 papers with them. So this is my collaborative work, and that's my background. As I told you, I'm a microscopist. I used to work with almost all type of microscopy, that's scanning and transmission electron microscope with fully equipped instruments with EDAX, EDAS, and EBSD and TKD, with all the modern facilities, with all the modern softwares, you can see. It's almost a separate and individual microscopist. That's my experience. You can also look into my YouTube channel, Dr. Vijay Asokan, where I have covered almost many topics, including microscopy basics and atomic scale approaches in electronic devices, machine learning, and the microscopy approach to visualize or image a coronavirus, such things, or basics of all the microscopy techniques, including EDS. It's covered here. So if you have a chance, just go and visit there. Apart from that, I'm also serving as a president here in Nortech. That's a Nordic Forum for Science and Technology, a Swedish NGO. And I have my own company in India, Mullai Academy International, and I am running a school in India. So that's my background about almost all of my expertise. Let's see why we need to go into microscopy and what are the needs of microscopy in terms of bioimaging or the biological field or biomedical field, we can say. So what are the need? Why do we need an advanced microscopy here? Look into the scale bar here, up to 100 nanometer. You can visualize elements or visualize the material or visualize the biological particles, biological organism through light microscopy. Let it be with frog egg or plant cell or animal cell or red blood cell or mitochondria or bacteria or flu virus. Then rest of that, if you want to look into a particle or an organism less than 100 nanometer, for example, new RNA type viruses, we cannot get all the information using light microscopy. Even though you can look or image viruses or bacteria through the optical microscopy or light microscopy, the detailing, a depth of focus, or the in-field magnification, you cannot get much information. You cannot derive much information using light microscopy. So that's allow us to develop an electron microscopy with an advancement into the field to get more and more, to derive more and more information from the particle or organism we are looking into. So that's the basic why we need electron microscopy. Of course, I will cover step by step with this information. Look into this. If you compare human red blood cell to the adenovirus or the polio virus, look into that. 10,000 times of reduction, or polio viruses covering 30 nanometer, and Ebola virus of 970 nanometer, or bacteriophage 225, adenovirus 90 nanometers. So our mosaic virus 250 cross 18 nanometers. So we need to develop a technique, in particular with an advancement in imaging, right? We need to derive as much as good and possible information to explore what we are looking into, to find what we need, to explain what we are imaged, right? So this is the background. Let's see, with a simple microscopy, our human eye, we have almost, we can explain the modern advanced microscopy through this cross-section image of human eye. Let me just take a laser point. That's simple to explain. So let's see this cornea and lens. This acts together as a single lens entity. And if you remember or recall the physics, the convex lens acts as it converges the light which it gets. So this is a simple convex lens which is introduced here. The light falls from the object and travels through the lens, get converged and focused at some point. This is a focal point. Right? The screen is called retina. And it sends a photoelectrochemical signal to the brain through optical nerves, and your image is processed. So there is a pupil and iris. This iris acts as a diaphragm, and this pupil which acts as a aperture. So what information we are getting from here is, you need an aperture to filter the light or the rays. Let it be in electron microscopy, it's electron, and in optical microscopy, it's light. And you need a lens to converge the light rays or the electron rays. You need a screen to get an image. You need some processing element to process, and you need some device to get the image information. Here it's brain. And the processing elements and the electronic signals in electron microscopy and photoelectrochemical signals in human eyes. So this is a simple concept, what we are getting step by step into a deeper understanding towards the microscopy first. So I want you to get understanding from the basics of microscopy. That's why I introduced in human eye. So this is how it looks. A light falls into a sunflower or a flower, and it goes into the lens system of human eyes. Then it gets inverted image in the screen. Then it processed through the optical nerves, and complete process done by the brain, human brain, and your image gets a completion of your image. So this is a simple system. Try to understand this. This is how electron microscope works. I will get step by step into that. So once your human eyes have some certain limitation in getting more broader resolution or deeper resolution and a higher magnification, there was a need to develop a system in particular to observe bacteria or microorganisms. So during 1590s, two Dutch spectacle makers, Jacques Janssen and his father Hans, started experimenting with the lenses and developed an early simple microscope with a magnification power of 6x to 10x. Later on, it was developed as a practical microscope by Antony van Leeuwenhoek, a Dutch scientist, using a single convex lens attached to a metal holder. If you can see on the left side of the screen, the metal holder, and was focused using a screw with a simple single convex lens. Later on, it was developed by Galileo, which was compound microscope, incorporates more than one lens so that the image is magnified by one lens can be further magnified by another. The lens closer to the object to be viewed is referred to as an objective lens, while the lens closer to the eye is called an eyepiece. So eyepiece and an objective lens. So here it's getting elaborated, like one lens which is getting image, which is further magnified by another lens. So we need a continuous or multiple lenses to get magnification. So this is a simple information we are deriving from the compound microscope. So this is a simple understanding. Then later on, developed theoretically, and the optical physics was developed by Ernst, Abbe, Carl Zeiss, Otto, and Koller, to develop further an advanced microscopy to image certain bacteria. So this is one of the microorganism which is imaged in optical microscope. So this is how it looks through the optical microscope. But we need also, in addition to that, what else we can visualize by optical microscope? Let's see. With the healthy blood, with the blood cells from the human body, you can identify or distinguish different types of blood cells, like whether it is anemia, low level of red blood cells, or leukemia, or sickle cell, or thalassemia, that's an abnormal hemoglobin production, or myeloma, or cancerous plasma cells. So these are the things we can distinguish from the human blood cells through the optical microscopy. Or then you can also visualize or derive information about the stage of developments of malaria parasites from the sample you're getting from different regions of your body, how it looks. So you can derive a life cycle of the malarial parasites, how it develops. So this is also like an optical microscope is also used to distinguish whether it's a male or female parasites. So it's also used to identify the different stage of development or growth. So this is how optical microscopy is used to get information from the malaria parasites. So let's see, this is another example of the malaria parasites which was got from South Sudan. So optical microscopy is it's developed further now to get as much as possible information. For example, this is the initial stage of imaging. Then later on there are algorithms which was developed from the conventional microscope to the advanced microscope in optical microscopy field to derive a color filter and derive much information. Even it is also from the example, it also shows a thyroid gland tissues, how it looks after eye smart, and the malaria infected human blood cells which get information from here towards an algorithm filtered imaging, then later on a color filtered image to get the spots highlighted by dotted rectangles indicate the presence of malarial infection, which was not actually not possible to get much information from the regular or conventional microscope, even though you can visualize or image the parasites, but you need to get more information. So the field is developed or advanced to get more to derive more information from the image what you get from conventional microscope through the softwares. Even it is slightly built up or developed, there are some image data sets, the data pre-processing. This is almost a computer based or software based or algorithm based, sometimes a modern programming which gets evaluated, then trained algorithm gives some prediction, the output with new image is formed. So this is like once the image is visualized or derived from the microscopy, there are new sets of work that's a post analysis process or image analysis process through the algorithms to get more information from the actual image of original microscopy images. So this is how the modern world is working on in respect of optical microscopy. But still, as I told you from the beginning of the slide, even though you get as much as possible information, but the resolution factor is limited in microscopy. You can get information from blood cells, you can get information from bacteria, but if you want to visualize a virus, this is not sufficient, even though with the modern softwares or modern algorithms. So we need a development, right? So let's see whether an advanced microscopy can fulfill that. This advanced microscopy is a little bit advanced than the conventional or traditional microscopy. So with this one, if you remember the compound microscopy which Galileo developed, one image which is derived from one lens is further magnified by another lens, right? In an advanced microscopy, there are further advancement with the introduction of different lens system, which the light source which falls into the aperture or falls or filtered by the aperture. If you just recall the human eye cross section, there was a pupil that acts as an aperture to filter the light rays which enter into the human lens. Similarly, the aperture system is introduced in front of all the lenses. The first lens which converge or condense your light beam, that's called a condenser lens, and lens system which is closer to your object is called objective lens, and lens system which project your image is projector lens. So this is a conceptual introduction or conceptual development in the advanced microscopy world. So you need a condenser lens, you need an objective lens, you need a projector lens. So this system is useful to get further magnification or the needed magnification at least. But still there is a theoretical limitation in an optical microscope, even though with any advancement, because that's from the physics perspective, because of the diffraction, because of the wavelength of light. The light ray microscopy uses the light which falls under the visible wavelength, which is around 300 nanometer, 700 nanometer, with a base calculation, and there are some physics calculation behind it, which limit the resolution factor towards 250 nanometer or 200 nanometer scale. So that's called an base diffraction. So with this one, you see, mammalian cell, bacterium, or mitochondria can be imaged through the optical microscope. But later on, with a microscope, with a virus or protein or small molecules, you need an advanced microscopy than this one, bypassing the limitation factor. Here, the limitation factor is a wavelength of light, a wavelength of source, which gives an diffraction. So you need to bypass that one. That's why we are going into an electron microscope. Why I introduced light microscopy in the beginning is, it's almost, the concept is similar. Here it's a light source, here is an electron source. But the condenser lens, subjective lens, and the projector lens system acts in a similar fashion as like any microscope. Even it relate to advanced light microscopy or conventional microscopy. So that's why the system is similar, but functionality is controlled by different parameters. Here is just a glass lens, here it's an electromagnetic lens. And it just transmits electron rays, here it transmits light rays. Here with an electron source and electron rays, which is converged through the condenser lens, but deflected by the scanning coils, then this is called scanning electron microscopy. Here it is transmitting, then it's a transmitting electron microscopy. So the concept is similar. The functionality is determined by different parameters when we compare light microscopy or transmission electron microscopy or scanning electron microscopy. So with this one, the electron rays, it's very simply developed from the cathode ray tube. You might have seen in your the old television, the CRT screen, there's a cathode ray tube. The cathode ray is nothing but electron rays. So the concept was developed as like it is, the electron rays can be deflected by an electrical field or the magnetic field. So electromagnetic lens system is introduced or the developed from the concept of cathode ray tube. So the electron gun, which is further developed from the cathode ray tube, it's a basic physics you might have studied in 10th or 11th or 12th standard, or in your bachelor degree if you studied physics. So the similar concept here. So the only way we are getting electron gun or electron source through this conceptual development from the cathode ray tube to the cathode ray which comes from the electron gun. So this is how the electron gun is now developed, and there are physics developed by the same period of time, which shows the electron gun can be deflected, electron ray can be deflected, and electron ray can be accelerated through the anode. So which gives information for the development of electron microscopy. Yeah, this is the older microscopy which I was operating in Indian Institute of Science when I was working 15 years back, and the modern microscopy system which comes like this. This is scanning electron microscopy with the electron gun. The electron gun produces electron rays. Those rays can be deflected by the anode, then enter into the condenser lens system, objective lens system, and then into the projector lens system to get magnified image. So this is how a scanning electron microscope looks. The detectors are here placed here with backscatter and scanning secondary electron detectors to get image. I don't want to go into much into the physics side, but I just want to introduce microscopy before going for imaging samples. So this is an advancement why we need an electron microscopy compared to optical microscopy. As I told you, the optical microscope can be used to image bacteria or parasites or the human blood cells. But we need an electron microscopy to get as much as possible more information. The example is shown here. For example, the Robert Hooke, the first microscopy developed by Robert Hooke in 1665, it shows a fly's foot. But scanning electron microscope image, which shows the same or similar fly's foot, which is imaged in 2006, which shows much information, and because of the depth of field, because of the resolution factor here. Similarly, if you take some cockroach or insects, the magnification factor through the electron microscopy brings as much as possible closer information with better resolution and better magnification compared to optical microscopy. So you need an electron microscopy in any sense compared to optical microscopy to get a better information from the source of sample you get. This is one of the bacterial imaged in scanning electron microscopy. That's a bacteria in human armpit, and this is a bacteria which is around the human hair, and this is a cholera bacteria which is imaged in scanning electron microscopy, and bacteria in your mobile. Wash your hands often. So that's a scanning electron microscopy image examples. I will just jump into transmission electron microscopy, which I was already explained, even though there's a filament which brings in electron rays. There's an anode which accelerates the electron. There are different condenser lens system. There are different objective lens system and projector lens system in transmission electron microscopy, where you get image in your viewing screen, and also there are some electronic signals processing to get your image better better after the softwares introduce certain things, and image can be derived as a computer files later on. So this is how the transmission electron microscopy looks. This is where the sample inserted, the sample holder looks like this. Here where you can place your samples. And these are the aperture strips, which I told you the aperture is placed before each lens. So the aperture shows how much electron rays can enter. You have a different scale of apertures, which shows how much electron rays can enter into the next stage. So this is one of my paper, which I was published with Pondicherry University with Professor Tangadurai, where they were used zinc selenide and zinc sulfide core shell nanorods. They used this core shell nanorods for bioimaging. I'm not good in explanation of biological things anyway. So they have a core shell nanorods which is around less than 10 nanometers. We want to identify whether selenide is in core or sulfur is in core. So we introduced the advanced microscopy. Of course, I have imaged this. We have an chemistem technology, which was one of the advancement in EDS in microscopy field, in Titan system with an atomic scale resolution. So I have gathered as much as possible information from HRTM images. If you look into HRTM images, you cannot differentiate whether it is selenide or sulfur is in core. You can get an image of core shell together. I try to get as much as possible information in a STEM imaging. Of course, there is a small brightness inside each of rods, which shows there is something inside the core, and that's separate from the shell. But we want to identify whether it is in selenide or sulfur. Then we introduced a chemistem technology. Then we get EDS image with selenide looks inside. That's inside the core, and sulfur which is placed in a shell. So zinc sulfide is in cell and zinc selenide is inside the core. This is what we derived. Then later on they have used it for manganese doped core shell nanorods into different processing to get cytotoxicity calculations, and then bioimaging for in vitro bioimaging, I think. So this is how we combine microscopy and bioimaging in nano field.